Hundreds of thousands of researchers around the world are working to improve life and address imminent threats to humanity. Often their research ends up in the scientific valley of death in the form of publications and patents that never see the light of day. That is about to change. Welcome to the Lab to Startup podcast, hosted by Naresh Sunkara, founder and executive director of the Berkeley Postdoc Entrepreneurship Program at the University of California, Berkeley. This show has two main goals: share the stories of those who have successfully founded startups based on their own research, and highlight resources needed to help those aspiring to launch startups in the deep tech space. Whether it's electric cars, vaccines, addressing climate change concerns, or possibly establishing life on other planets, Naresh and his esteemed guests want to help scientists, engineers, faculty, and researchers bring their innovations to market. Learn more and subscribe today at labtostartup.com. And now, here's Naresh.
My guest today is Marija Vrljic, the co-founder and CEO of Scalmibio, an early-stage biotechnology startup that developed a transformational technology focused on oncology applications. The technology converts therapeutic antibodies into prodrugs activated specifically within a tumor microenvironment. Such restriction enables their broader use in combination treatments, higher therapeutic doses, and limit side effects. Marija received her Ph.D. in postdoctoral training from Stanford University. She was recruited as one of the first scientists at Impossible Foods, later by Eve Biomedical. Marija's startup journey is a super inspiring story because the idea for the startup originated from her own fight against thyroid cancer, as she felt a need for better drugs with lesser side effects and better efficiency. Marija did not raise any money from angels or venture capitalists. She got some non-dilutive grant money from NIH and developed the super impressive technology at an incubator in the Bay Area, which led to them being acquired by ALX Oncology in October of 2020. I'm super confident that you'll enjoy my conversation with Marija. There are so many lessons for those considering startups. Also, as you know, from time to time, we bring on early-stage deep tech founders to pitch about their startup. Today, we have Hunter McDaniel, the founder and CEO of Ubiquity, an advanced materials company powering product innovations in agriculture, clean energy, and security. So please stay tuned to the end of the show. Welcome to the show, Marija. When I first heard about you in a casual conversation from our mutual friend, Doug Crawford, in passing, one thing that stuck about you was you being a solo founder who took the company to exit. And that was when I decided we need to bring you on to this show as a guest. And thank you for accepting the invitation to share the story. I heard even more inspiring things from you during the introductory call. So I'm really looking forward to this conversation with you.
Pleasure to be here, Naresh, and thank you for the warm introduction. And I'm really kind of happy to be a part of this and try to share my experiences with graduate students and postdocs, and hopefully to kind of give you extra wind in your own sails to start a company. And just to correct Naresh, I am officially not a solo founder. I was a solo operational founder, but I do have a co-founder.
Officially operational. That's interesting. We'll get into that. There are so many topics I could start this conversation with, but I think it makes the most sense to start with the inspiration and the reason to launch Scalmibio, as that is a thread that connects a number of other stories and experiences in your life. So let's jump in there. Tell us what Scalmibio is about.
So Scalmibio is oncology-focused company. Our mission is to make antibody therapeutics safer and therefore more effective. It was inspired by my personal story. I got diagnosed with a thyroid cancer around 2016. I did not know that I had it. It took around between diagnosis, realizing something was wrong, having a surgery, doing a post-surgery treatment. It took around 6+ months. And during that time, I went through the whole process as a patient. I also read a lot about the current state of the oncology therapies, both the biologics, especially antibody therapeutics. And what really stuck with me, given my experience as a patient, was the safety profile of a thyroid cancer treatment. You get the surgery, resection, and then for certain subtypes, you can get this radioactive iodine treatment, which is a very targeted, very selective, with minimal side effects. And that experience contrasted with the experience of other patients who primarily go through chemotherapy really inspired me to try to apply and create essentially antibody prodrugs and fit in within that space to bring this safer treatments, but yet very, very effective to a larger fraction of patients for more indications. So that's kind of how the company started.
Wow. It started with your own journey of cancer treatments. And let's stick to first principles. What are antibody prodrugs?
As you think, for example, a long time ago, there are certain both the small molecules, large molecules, there are the whole pharma industry and our way of thinking about any prodrug, be it a small molecule, for example, even like aspirin is a small molecule prodrug, it needs to get converted in your body into its active form. And that's something that you take on a daily basis. And then from the perspective of the antibody prodrugs, I think folks around like 10, 15 years ago, they're like, for example, they've been trying to make peptides linked to some polymers, and then they get processed in the body. In that way, your body converts something that's in its inactive form or less active form into an active form. And then this whole concept got also translated into biologics, and in particular, in our case, in the antibodies. And we're not the only ones in this field. It's a very active field with the idea that potentially, for example, antibodies cannot differentiate between targets in a normal tissue and in a tumor tissue. And there are so many different targets that are, let's say, expressed at the equivalent levels between tumor and normal tissues that you could try and use. Then if you could convert antibodies such that, for example, it's inactive while running around your normal tissues, and then it becomes active within a tumor tissue, you can increase what's people in a field called therapeutic index. So you can increase the difference at what you can dose and between your side effects that are, for example, dose limiting. So in that perspective, the antibody prodrug is philosophy-based, the same prodrug as any other, let's say, small molecule drug or a peptide drug or any other protein-based thing that you want to process within your body to create an active form.
Okay, Marija, let's dig a bit deeper into the concept of prodrugs when it comes to antibody treatments. Can you elaborate on that subject, please?
Antibodies are designed and they work such that they bind to a specific target or antigen within your own body. And those can be anywhere. They can be a normal tissue, they can be a tumor tissue. And they're very powerful drugs. However, there are very few targets that are overexpressed specifically in tumor tissue. And people have worked on those for a long time. And now you have this universe of targets that are expressed almost, let's say, at equivalent levels between normal and tumor tissues. And how do you access those? Because if your antibody is going to bind to them, you're going to have a side effect. And sometimes they're severe enough that, for example, this potential therapeutic cannot go forward, either in clinical trials or even preclinically, because the side effects are too severe. And then the concept of the prodrug comes from how do you make it inert up to a point where you want it to act. For example, in a tumor tissue, or if you have autoimmunity in autoimmune kind of affected tissue. So the whole thing is you give it in a version where it's inactive, and then your own body somewhere, somehow converts it into its active form. And it's a very active form of research. Concept-wise, it's a very old concept. It's been applied over all kinds of therapeutic classes, small molecules, polymer drugs, imaging drugs. If we use it in research on an everyday basis, like when you, for example, you want to label cells intracellular, you give it fluorophore that crosses the membrane, gets processed, gets trapped inside the cell, and it no longer it's going to come out. Effectively, it's the same as a prodrug. So that's kind of the concept where we want to do. It's another way of how you make a therapeutic act in a tissue that you want it to act. If you can find a mechanism, so you can convert it at a local focal spot of action.
Thank you for explaining that. Was there like a unique insight or a research finding that compelled you to launch a startup around this concept?
So again, kind of going back to what you do when you become a cancer patient. So I said I was reading around and what is latest, what is greatest in the oncology field, and the antibody therapeutics really caught my attention. I think that was around the time also when you think about it, but immuno-oncology really took off, it was really now widely accepted. I remember from my graduate school days, I have a friend, she was in John Levy's lab at Stanford. So just using the whole concept of using your immune system to fight cancer has been really old in concept. But then right around on the wings of it and kind of getting the more clinical attention were these immuno-oncology findings, for example, from Jim Allison's lab with anti-CTLA-4 antibody that did really, really well and kind of opened up the whole field at the time. And at the same time, I also kind of came across and was really particular about, for example, CTLA-4, since it was kind of inspirational molecule at the time for me, was that it was very efficacious, but it also had kind of significant side effects that if you could turn it into a prodrug, then effectively you could potentially minimize those. And it wasn't a concept that, let's say, we came up with. Actually, there's a company out there called CytomX. They're absolute leader in the field of the antibody prodrugs. And they had the same process going on and concept with, and a collaboration with Bristol Myers Squibb, who was commercializing the antibody originally characterized and kind of the concept mechanism of action by Jim Allison. And by looking at different antibodies, and I think the crucial insight for us came from, given my background, I spent some time in the structural biology lab with Axel Brunger and thinking about from this biophysical perspective about proteins, how they're folded, their conformational states, isoforms, things of that. So both for my graduate school work and some of my postdoctoral work, I now kind of like played around. And there was this insight that the antibodies are folded in a very conservative fashion, and kind of looking as to what CytomX that they were inspiration for this company. Absolutely. I've seen that, and the way their tech works is effectively you need to have, they design it such that you have a specific peptide that binds to any specific antibody. So effectively you have a one solution for one antibody. And I think kind of the way companies started was by this realization that the antibodies actually have a conserved fold that you could take their N-termini, which are hovering around the antigen binding site. And if you were to connect them, you would create this effectively a steric hindrance. But you can connect them by, for example, a polypeptide or a peptide, and you will create steric hindrance that kind of hovers over the antigen binding site and therefore physically prevents, sterically prevents binding to the antigens. And what our thought was at the time that because there is no covalent interaction between this peptide that's hovering around the antigen binding site and the antibody, that you can apply it, if this could work experimentally, that you could apply it very quickly to several or multiple different antibodies, regardless of the sequence or the species. So that was the motivation and kind of, hey, this could be something that maybe can help R&D, that can help this process of conversion of any antibody into its prodrug form. So that was, I think, that kind of made the crucial thing, kind of just sitting there and scrolling through the different antibody structures publicly available in a PDB database. It soon dawned, like combining these two things together, there is how CytomX did their approach with understanding that if you could connect the two, that the particular parts of the antibodies are folded such, they have this unique conservative fold. And if you connect the two parts together, that you generate this almost like a peptide or a polypeptide based steric hindrance that does not interact with the antibody itself. So in a way, it offers blocking without interacting with the antibody. And then because of that feature, if it were to work experimentally, you could apply it very quickly to any antibody, regardless of its sequence, regardless of its species. So effectively having that approach that CytomX did, you could really speed it up, parallelize it, apply it to many more molecules faster. And I think that was the motivational or like really inspirational idea. If we could get this to work, if this does work, then maybe people will be interested in that. And then I think kind of another part that went into it is this concept of using a proteases as to release anything and activate a prodrug within a tumor is something that clicked with me since I've seen the work from graduate school times. And at the time, I kind of thought was like, oh, it was a neat idea. Not quite sure what the application is going to be, from Roger Chan's lab from UC San Diego, where they designed a peptide labeled with two fluorophores such that when it's in a one folded conformational state, it's actually quenched. And when it gets clipped by a proteases within your own body on a tumor site, these two fluorophores, there is kind of cell-penetrating peptides, they start fluorescing. And they wanted to use it to image tumor boundaries for kind of like the fluorescence imaging guided surgeries. And it's been now, maybe 15 years. That idea from the Roger Chen's lab got turned into Avella's Biosciences. And then just very recently, they had after this whole idea happened, they have actually breakthrough designation from an FDA. So I believe that this kind of building these two concepts together, understanding that it could work from both aspects. So that's, I think, how things started.
Wonderful. Thank you so much. You bring up a bunch of good points for the audience to take away as well. Innovation in deep tech comes from a lot of failures in the past, and scientists like yourself observing and taking notes of these things, what didn't work 10 or 15 years ago might be working now because of technological advances and unique insights by scientists like yourself. Oh, I've taken this protein clipping there, and then maybe I can use it to clip these prodrugs to make them active in the current state. And that is what investors are looking for. What didn't work previously, and what is that unique insight that you bring to the table that will make this startup a success? That's what builds confidence in you as founders. Is there anything else to add on that subject, Marija?
I think that, no, Naresh, that's a very, very good summary.
Who was the first person you took this idea to, and what did that conversation look like, and where did that end in that aha moment where, I need to start a company? There is some meat in this. Walk us through the decision-making process.
First, I was still poking around, and I talked to several people who had similar backgrounds that I, let's say, knew from postdoc. I kind of also wanted to talk to people who had therapeutic experience. That's kind of through my own personal network and knowing folks. The main person I talked to was Jaume Pons. He used to run a company called Rinat that was purchased by then Pfizer. He was the CSO of that company. At the time, he was also a part of a venture capital firm called VentBio. And then he also founded his own company, and he was kind of running it. So he had a really, really good experience. He saw several antibody therapeutics go through from the R&D stage all the way through clinical trials. And he was someone that I really turned to talk to and say, hey, I had this idea. This is how it works. This is the background. This is how it's different from, for example, from the current competition. And then just kind of based on a piece of paper, if we could see experimentally, just very basic proof of concept, this working, what do you think? Would this have a leg to become a company? And kind of through a series of conversations with him, and he became a co-founder effectively.
So we kind of watched it. Non-operational.
Correct.
Co-founder.
He's my non-operational co-founder. So there was lots of conversations back and forth and kind of fleshing it out. And that led to effectively deciding, okay, let's see if this has any experimental legs to stand on. And then let's go and make a founding of the company.
Thanks for correcting me on the non-operational co-founder having one. What does a non-operational co-founder mean, and how did it work out for you? What was the role this co-founder was playing in the evolution of the company? If you can elaborate on that, that'll be very helpful.
So non-operational founder, I think the way I use that term, and I think the way people, when you talk with them, kind of try to imply the term, is a person who's not there 100% of their time. Effectively, a person who is either they're a part-time, or who effectively does not run the company on a daily basis. It does not participate in the running of the company on a daily basis. So if you want to make a comparison, it's very similar if you were to have a faculty co-founder, and then they run their lab, they have obligations there, and they still have a significant input into your company or opinion on perspectives, but they're not there physically. And this was a very similar relationship in that front. I met Jaume and through my network, and he was someone who had tremendous and very unique experience of seeing therapeutics, antibody therapeutics, go anywhere from the benchtop that he himself worked with his hands all the way through clinical trials. He chaperoned more than 10 programs through phase 1, phase 2, phase 3 clinical trials. He was CSO of Rinat, a small biotech company that was functioning under Pfizer. After he left, he founded his own company. So he was someone who's seen a lot and the whole process, and had a really kind of unique window into how things work. And the way our relationship worked, from the first time I had idea on a paper, I talked to some other folks that I knew, and then I decided to just kind of a very basic thing, if I can add, if you were to have this kind of a product, for example, if you can add a deadly key to an antibody, would you be interested in having this? Or would you, how would you think like your, kind of, what would your perspective be? And then I approached him and we talked and fleshed it out. And like there was lots of conversation, lots of back and forth perspectives, like if you want to call it like a strategy sessions, kind of what would people be interested in? What would you need to go try raising money? Like even from the very, very basic operational things, like, okay, I have this, we have this design, or what target do we want to go apply this to? Which antibody to focus on? What would we need to have? Or even just discussing experimental data. Hey, this is what I got. What do you think? If these are the next steps, what do you think about them? Does it make sense? Does it not make sense? So it was a very active and then dynamic relationship. And he was always available effectively. If I had to talk to him, I would call him, text him, and we would talk that same day. That was always something I very much appreciated. But because of his very other obligations and interests, I was very grateful for the time that he had for me and perspective and kind of that he offered, but not operationally. And then just to kind of give you another heads up, like I think some of when you talk to people, potential investors, like potential angel investors or seed stage funds, and people are very ambivalent as to how they perceive this kind of situation. I think some people say, hey, you do need to have another co-founder who is 100% operational with you. Like effectively, you need to have kind of all, like both of your hands on the deck all the time. And that's what we want to see, like effectively, that's what they were looking for. So you have to kind of, I think a part of this lesson for me, or what I've learned, is that we've got to have folks who have lots of experience, who with the perspective, who can help you have a perspective and kind of fill up all these gaps that you have in your own understanding of the ecosystem, understanding of where the projects needs to go, what needs to happen. But you also have to have folks who have who can be there and have a will and the ability. And that, by I mean ability, you need to have it's really early in early stage startup, very early stage, before when you're still like financially struggling, you've got to have people who can take that financial risk. And not everyone can do that. Like, if you have, it just kind of depends on your personal and your family situation of whether a startup, early stage startup before like solid funding and with short runways is something that you can afford to do effectively.
And what happened next? Like, were people lining up with money with you? Cool idea. You have a nice co-founder. What happened next? We would like to get both the good sides and the bads. I think the trials and tribulations, where people jumping up on the idea in terms of funding and giving you lab space and so on.
Let me kind of go back and forth on this. I think that question becomes, at what point, like how much do you have to go get money from people? That was different about our startup compared to, let's say, other startups. So let me just kind of like from my personal observations and what I've seen, like as a graduate student, probably lots of your postdocs and grad students go through this. You see, let's say, colleagues from different labs, maybe even your own PI labs, they worked on a project in the lab, they compiled some data based on your NIH grants, you kind of work on this project and you're like, oh, hey, this can be a product. So you talk to your PI, you talk to your tech transfer office, and you try to figure out what's going on, but you have some data package. And I know like probably by now, and then that was like much later than when I was in grad school and postdoc. So just as a tip, go and take use of those services. For example, I think Stanford has a program, probably Berkeley as well, where now they have an eye on what do you need to go and like what kind of data set do you need to build for folks with funding to be interested in investing. How can you de-risk it enough and make it interesting enough for them to say, hey, this is a competitive idea that you can take forward. There is that package going straight directly from the academia. Then there's a package, I think, from folks who already, what people call serial entrepreneurs. They started some companies before, they had successful raises, successful exits. Effectively, they had a proven record that, whatever they can come up with, or let's say a license out of academia, they can turn into a functional product that someone wants. And I think every investor has this calculation is this worth it for me? Can I get a return on my money for this particular investment? And they make, I think my personal impression is they make a different matrix calculation as to what does the risking mean for them. Is it what they call the team, which is is it folks who have gone through this path before? Or do we have a good enough? Is this data package really compelling? Is it really competitive right now? And folks that this company can sell product to would be interested in this space. That's something kind of that we try to make lots of calculations with. And to be honest, I did not really know where on that spectrum we would to land. And then there's also this calculation of, is it too early? Do you have enough data? What % of the company would you have to give away? Do you have enough data to attract someone's attention? For some people, you can say, I have the idea on a paper, and because it's either a compelling idea or I already have a proven record that I can sell this and make a viable product out of it that can make money for folks. Maybe, the bar for having a data package is much lower. And for us, that was a continuing scheme, what's good enough? Is it good enough to have just basic in vitro biochemistry data? Is it good enough to have some cell assay data from protein level? If you're coming from a structural biology lab, sometimes okay, I have this characterized protein stuff. Is this enough? Or do I have to have some in vitro cell assay data? Is that enough? Or do I have to have in vivo mouse model data? Is that enough? And through that series of conversations, I think I learned that the standard effectively for where we were is effectively you have to have an in vivo model. Otherwise, people are very reluctant and competition is so high that they were not as ready to jump in. They kind of ask, you need a proof of concept in mice, then come back and talk to us. I did try to go out, and I tried going out talking to, let's say, angel investors. And I also tried, for example, applying to Y Combinator. But they tell you up front, they do not really like if you don't have at least 200% co-founders. So some people are very upfront about what they believe in. Other people don't care, but they just care about other matrix points. For example, what was, I think, going back, even though no co-founder, this is more like a funding question, that what ended up being early on confusing to me is that very different check price points wanted to have the same data package based on maybe who I was, for example, or who we were at the time. You go to angel investors, they hear your story, they're interested, but they say, hey, this is great. However, it's a therapeutic, it's a long-term investment return for me. It's not something I can see my money back in 2 or 3 years. It's going to be a long haul. It's a high risk. Therefore, I need you to have a proof of concept in mice. You go to informally talk to a seed stage guys. They effectively almost tell you the same story, very similar story. I need to see a proof of concept, proof of efficacy concept. And ideally, because you're having the safety premise, what our technology is promising to make antibodies safer. So you've got to have they want to see a safety and efficacy readout. And then you go to Series A investors and they kind of tell you the same thing. So and then how do you resolve this space? Kind of doing some research and maybe through talking and then through talking other people who have gotten funding from those groups, what do they have? How decisions were made in their case, I think helps you understand the ecosystem and maybe kind of helps you apply to understand where these different folks or groups or finance groups are coming from. And at that time, I think for the early stage investors, say potential angels, I think what I heard most often is that if they don't know you personally, if you don't have a history of making money for someone else or for them personally, then their bar for data package and them having a comfortable comfort zone that they have de-risked their investment is much higher. And it's something that you can learn only by talking to them. It may not be for every industry, it may not be for every company. If you're a device, if you are something else, it could be very different. It's just the only way to figure this out is to go out and talk to them and see, kind of what pushes their button in a positive or negative way.
So you figured that you needed some in vivo model. That was your starting point, that this is one milestone where I might be able to be funded.
Yeah. The only way to figure that out is effectively by talking to people. And when they call this informally, you try to kind of go through again, your network and ask some people maybe who are in the VC space, or maybe who've been through this, who have similar companies, and you say, hey, I have this. This is my situation. What do you think? And you have to always balance this out with, in academia, you're always so open and you can talk about any aspect of your research at any point in time. That's the academic culture. But now that you're in company, you've got to make this decision. Is this ready for sharing, or is this still considered proprietary? And proprietary in a confidential way, or it's always proprietary, but is it non-confidential or confidential information share? And I think when you are starting early and you don't have necessarily all of your patents filed out, because even that is kind of significant startup cost, you have to make those balancing decisions. And I had like a third person who really kind of jumped into this, and I knew him from postdoc time. And he right now is a professor at University of Colorado. He also has his own company called Omics Technologies. And I think kind of like with him, we kind of talked through and decided to go SBIR route through the National Cancer Institute. That was really, really helpful for us to go and search this kind of government-based funding, no strings attached. It really also just whole writing a proposal helps you kind of flesh out your concept, your business plan, the funding. I think it's like really, really useful exercise for a person who hasn't done this process before, just to kind of how you think about the budget. The SBIR program really makes you focused, makes you make a budget, really plan it out, think about how am I going to go spend this money over the next 1 or 2 years. And then people get to comment on it. Sometimes comments are harsh. Sometimes they're really nice. Sometimes they're like wide range of it.
How about you? Did you get the funding the first time around when you applied for SBIR? By the way, SBIR is a phenomenal way of being funded. It's non-dilutive, proof of concept funding for anybody who's trying to get the startup going on.
Yes. And then just to get a plug for the SBIR programs, this was not available. These are new programs right now going on. So for your listeners going forward, and maybe to go to your tech transfer office and figure this out, SBIR has these classical programs like phase 1, SBIR, and what they call STTR, which is transfer from academic institution into a business that you could work. Let's say if you want to take your own project to take it with you, or SBIR, you just apply as anyone's idea as a small business. But now this year, they launched 2 new fantastic programs that Naresh can maybe like have an extra session on this. So one is this transition. It's STTR like for people up to, I think, 7 years of postdoc, post-graduation, and it's up to $2 million for taking your own idea. This year was the first batch. You take your own idea from your postdoc or graduate school research and you apply it, you write a proposal, and it's a 2-tier. So there is a phase 1. And if your milestones from phase 1 are successful, it turns into a phase 2. That's where this goes to like almost $2 million. Your first tranche is like, let's say 300,000 to 400,000. And it's an absolutely fantastic way to have alternative career to a career in academia. And it just started this year. So please go inform yourself, read about it, go for it.
I will have some plugins in the show notes for sure, but I'll try to bring somebody from NIH to talk about that program.
Yeah. And then there's another program. I'm not done yet. The NIH is really on top of it this year. There's this other program, which is, I forgot its name, but it's meant as a complete pilot. So you do not even have to have a preliminary data for it. For SBIR and STTR classic grants, you've got to have a preliminary data. They have this new thing, again, launched this year, and it's a truly proof of concept. You can have innovative idea, no preliminary data required, as long as you write good proposal and has a good scientific rationale. And both of those, I would think, are really, really game changers, fantastic things. And then you have like regular SBIR. So you have like 3 options to really get yourself organized and get non-dilutive funding and really help you start.
I'll reach out to you after the show to get some notes on that. But let's come back to your story. So you wrote your first SBIR and you got funded the first time around.
Yeah, we got funded first time around.
Imagine and how big was that check?
That was for $300,000. And let me tell you something else what we got. According to our program officer, that was the highest scored grant in her portfolio.
Congratulations on that. You don't get to hear that.
Thank you. And I have to kind of give credit to Kirk Hansen at that point, because due to his grant, I never wrote a grant before that. So that's another thing where you really having him, and we were co-PIs in that grant, as an omics, so not as a professor at an academic institution, but actually that was a consortium with the small company that he founded. Omics Technologies, which is a proteomics and metabolomics company. And we had this joint program together. And what really helped was talking things through with him and having his guidance on how this grant needs to look like. Because if you never wrote a grant, and now again, sorry, I'm now sounding like an advertising board for SBIR. At the time, they didn't have this program, but now they actually have writing assistance. So you can apply before you submit your SBIR or STTR grant, and they have this mentorship program. They will help you write their grant. They will offer the criticisms, suggestions, how to improve different sections, what you can improve writing on. And I think when I've heard from some of the other folks who got it after us, it's really helpful. And it's only for, I think, first time people applying. So again, take advantage of that too.
Yes. I will definitely have the link on the show notes for sure.
Yeah. So that was really helpful for us. It was really a game changer for us. That helped us. We got the grant. We had a kind of relatively big check in our bank account. Sorry, it doesn't work like that. So the way that grant works, you do the work, you apply for reimbursement, you get reimbursement. But just having that there really helped us move forward. And around at that same time, we were also looking for a lab space. And that's kind of in the Bay Area. We just looked through different, asking from some network folks, can I sublease? I got this money now, I have funding, can I sublease from you? And lots of times we heard a no, and not from the people from small startups, but actually the landlords would say we can't. And I think there's some kind of law in California that subleasing is really difficult. So that was really challenging. And then incubators are really also they have a long wait list. And that's where actually NBC really came to the rescue and really help us really get going by actually having a space for us. So at the time, we just rented one bench and access to their shared equipment. And it was I was so happy when they said yes. I was on the seventh heaven, as they say.
Doug mentioned that. What stood out from this experience just up till getting the grant was the network or the relations you maintained with other postdocs in academia, the Kirk in this case, I guess, and willing to work with other people. It's so important. And one of the things that stands out is you are pretty open about talking about the idea to other people to get the right feedback. We tell our audience that ideas are a dime a dozen. If somebody can just listen to your idea and form a company, maybe there's no IP there. It's hard. Execution is hard. But what were you thinking? When you're talking about the idea, were you worried that somebody will steal the idea because this is not something you had concrete? It was just an idea at that point.
I was concerned about that because to me, it seemed like a very elegant and kind of simple idea. And simple in the sense that I had ideas about companies before. And lots of them, it's been something I've been actually thinking about. Sorry, I didn't say this. It didn't just occur to me hey, I want to start a company. I've been kind of musing in the background while working at different startups since my late postdoc times. And some of these ideas that I had, as you said, execution-wise, they seem pretty challenging. So I kind of kicked them to the curb at the time, just because I felt like it's going to take too long and it's too experimentally risky. And what I really liked about this one was that it felt experimentally reasonably straightforward. And for that reason, I was, as you said, a little reluctant to just really talk about it openly until we filed a patent. So that's something else that everyone tells you, you've got to file a provisional patent before you go talk to anyone. And we've done that. Apart from the only exception I made was Jaume. I decided to just trust him and talk to him. But until that point, after that, I only talked to people after we had provisional in place. And that's the advice that you will get all the time, that you've got. Maybe as you said, you can flush very basic idea. Hey, if I make this antibody prodrug, if I can make it such that you can attach this leaky to any antibody, and would you be interested in funding this? You can talk about it in that way, but not exactly. But lots of people, it's a hard thing to do, because when you can sit down and talk to people with scientific background, they're like, hey, but how are you going to go do this? And there's this other company doing X, Y, and Z. So how are you different? So you have to be able to be flexible on what you are comfortable sharing.
Absolutely.
At any point in time.
Were there naysayers who would say this wouldn't work? Did you have that experience? How did you deal with that?
I think people said, not that it wouldn't work on our idea. I think what people were somewhat reluctant on is that whether the whole field would work. And that's something really hard to counteract as a small startup. because they would say there's no clinical proof of concept, which is true. There's no biologic pro-drug, or let's say antibody pro-drug, yet approved on the market with a prescription to patients. There are things in clinical trials, but they have not crossed the final line yet. And having that skepticism is something that was kind of disheartening, more from the perspective like you can't do anything about it. We are so early on, you're such a small company, and effectively you're relying on your competitors to cross that line. But also, you know that line is so far out. It's like, what do you do? Like, it's not like that line's going to get crossed tomorrow. That line's going to cross, I think, 4, 5, 6+ years down the line. And then what do? Do you just fold and walk home, or do you continue and take a risk? And that's something that you have to make a decision on. Like, and I think that kind of also helps with talking to people, like what actually are they looking for? I think people talk a lot about it, like these milestones and what's going to be a turning point and what people are willing to invest more cash into your company or your idea. And understanding where that point is for different in the field is useful as well. And I think the other part that, let's say, NBC has very useful, they have this partnership program they call the Golden Ticket programs. They have partnership with different biotech and pharma companies, and almost all of the partners, and you probably heard that from Doug, runs what's called a golden ticket. So effectively, you submit them your non-confidential deck, and that particular partner awards effectively a rent. They pay your one bench rental for 1 year, which is not much. We're talking about maybe $20,000, $30,000 a year, which is for them is insignificant. But even if and I didn't get any one of those, just to kind of let everyone know. But what was useful, I did get to talk to people. So as a part of the application process, some of them do select, let's say, a smaller number of people that they talk to. And then you hear things like that, like, where does their reluctance come from, if any? And that's really good to hear.
That feedback loop is super, super important for early stage entrepreneurs, because we have some assumptions and there are things that we haven't tried and we're just blindly moving forward in the hope that it'll work out. But getting some feedback is really important, I feel. Thank you for that, like insights into that. Like, so for the sake of my sake and the audience sake, give us a sense of the timelines. So when did the idea start? When did it get funded? And what happened after that? This is 2021.
So we had a very, very slow start. And let me just kind of preface it, because we kind of decided to bootstrap it on ourselves. We founded a company in 2017, mid-2017. We got SBIR funding in early 2019. And then we did apply for one more SBIR grant in a different aspect, and we didn't get that one. So that was one really interesting experience after the first one. And then effectively, I was just decided not to go out and fundraise. I was just effectively relying on our own bootstrapping and SBIR for the grant that we were awarded to get to that in vivo animal model. Once we got the data, we got this data in January of 2021, so just this year. And then things really started rolling at that point, as predicted. As people told me, once you have mouse data, come back and talk to us. And the interest really did change. So, what they tell you, it's kind of actually true.
This is pretty fast, I guess. The ideation till you got your SBIR is just 2 years, but it's a tough one. But you got your mouse data by January of 2021. And was there a disease model that you were pursuing, or is it the concept that you're trying to prove, the proof of concept?
Oh, it was a proof of concept. So we got an SBIR grant on making a CTLA-4 prodrug. So once you kind of commit to that particular target, there are mouse models that one kind of has to go into. And the reason why we kind of chose that path, as I said, initially, that was a very interesting to us. But in addition to that, like a mechanism of action, thanks to Jim Allison and the folks, it's really well characterized. So you know what you're looking for, you know what your readouts are. You can make a very reasonably straightforward comparison, like how does your molecule compare to a positive control?
Nice.
For us, that was really, for a small company, kind of having that framework was really helpful and enabled us to have a proof of concept. So that was really kind of very good synergy for us.
Great. And then I think this is public knowledge. We hear that a company got acquired by ALX Oncology.
Yes. So that is a public knowledge that happened just this past October. Thank you. And we decided to go down that route as the ALX has a very experienced clinical team. And given where we were if our stuff is going to reach the patients, I think this is the fastest way to get there. And we hope we can get there, and then we won't.
Walk us through that decision-making process. Like, you could have gone out and raised a lot of VC money, like a lot of other biotech companies do. And this was the other route. And did they find you, or did you have to find partners? Like, what is that process like?
I think that process was two ways. We were looking at how do people raise money? And there are a few ways that people raise money in the biotech world. And if you just kind of look a little bit around, you'll see the pattern. And also it's part through this, let's say, also concept and idea that, let's say, MBC Biolabs is fostering by having these biotech partners for themselves and kind of opening those doors to different companies within their ecosystem. And I think idea is there are two ways to fund yourself in the biotech world. One is just to solely rely on a VC funding and do raises until you go IPO. And that's usually another way you raise money to kind of push your clinical programs forward. And then the other or parallel way or complementary way is this making partnerships with different biotech and pharma companies. And if you kind of like look around, like both models are very actively pursued, at least from my perspective, by a series of companies. So we always kind of had, I think, the lookout is like which way one can go. And there's really, really successful stories. And probably through Naresh, you went through folks who had these like really successful partnerships with bigger companies, and they've done really well as an outcome for themselves. And there are some stories that did not work out. There's always like a rainbow and continuum of experience. This is like a different plug that I really want to put out. Like, people tell you about their stories, but for every positive story, there's a negative story. There is a middle story. So you really have to just take everything and just to collect different experiences and never really get set into like there is a way from point A to point B. Kind of starting a company is almost like a research. You never really have, there is no direct path for everyone to follow. There's always these different curvy roads, and you have to find your own, and talking to folks can kind of help you maybe get inspired to try kind of road B, road A. It's very, very similar to research. I think you have to have the same resilience and mindset and know that in a way you're on your own a little bit, but you also, there's this whole thing that you can lean on and you can get their experiences, and maybe some of those patterns work for you and others don't. And it's kind of in a similar fashion here. We're always kind of like feeling both ways out. It wasn't always way one and way two. It was always kind of both and see what would really make a difference. Because the way I thought about this, our technology before, that was another, let's say, step that I went through. I didn't talk about it before. First, I thought it would be a great platform. When you kind of think about it, in its very essence, what we've developed is literally the liquid that you add to an antibody, and it turns it into a prodrug. That was our goal. And originally, you didn't really care what antibody that would go on. My original thought was like, hey, like these folks from bigger companies, they have extensive pipelines. They know what failed in a clinic. Potentially, for example, they have really targets that either preclinically or clinically failed due to the safety issues. Maybe they're interested in resurrecting them if they had a way of making them safer. So that was actually my really, really original way of thinking of how to approach potential partnerships and how to spin this. I thought that was like the crux core value of this, of our technology. Another thing that I've learned that I didn't mention before was that people really look for products. You have to have a product, and then the product is backed by a platform. And it took me a bit of time by talking to these different folks that we talked about to understand that that is another like essentially standard. That's what they expect to have. You have to have your really lead product, one that this is your flagship going forward. And then you have this potential of a backup. It doesn't work without having a lead. So you have to yourself pick a lead for yourself. I think kind of synthesizing all of that together, we always try to keep everything together. And then that kind of the reason why, let's say in this particular way, not going down the VC route, when you kind of think about it, I really wanted something, and I kind of like alluded that to before, what's the fastest way into the clinical trial towards the patients? And I made a call, or we made jointly a call, that raising VC funding in these like kind of consecutive steps and going through a process of hiring, doing all of our own hiring and assembling the team that spans from R&D team, development, clinical team, it's going to take too long. I thought that the fastest way to move forward is to do what we've done, get into a team that's already there, that has a really good track record of being very successful themselves. And I thought that if anything, they could really give the best chance for our programs to succeed.
You bring up so many good points in there. So one is there's no direct path from A to B or A to Z for most startups. You have to figure it out by yourself. And one of the goals for this podcast has been like, just at least bring various experiences, tell people how they navigated this path. It may not be the same, but it's just exploratory process, the thinking process that might help people. And the other important point you brought about was, if you're trying to build a platform, you definitely need to have that lead product. I hear these pitches all the time. Oh, we're a platform play, but you got to have that product. Nobody's going to trust you unless you have that. And the in vivo model, I think that is going to be a stop point for most startups that I'm hearing that repeats again and again. Thank you for all those inputs in there. Let's slightly shift gears. Let's take you back to your grad school and postdoc. You work with some superstar advisors at Stanford for your PhD. Hardin McConnell, who won President's National Medal of Science, and the Nobel laureate William Morner. And your postdoc was equally prestigious with advisors Axel Brunger and Nobel laureate Stephen Chu, who was the Secretary of Energy for US. What was the experience like working with all these superstars? And did they convince you to go into academia, or did that whole working with them not drive you into academia?
Thank you for that summary. And I think what I can tell about all of my advisors is how fearless they are, how they're willing to open doors, how they're so curious about different fields. And I think from that I learned from all four of them, but especially from Hardin McConnell, since he kind of shaped me as a graduate student along with W. Murner. What does it mean to do interdisciplinary work? And if you remember, like I've done that 15 to 20 years ago, when people were not as open-minded, I think, or not as used to interdisciplinary work as they are right now, how you have this flow of tools, skills, how do you build that whole process? I was focused on imaging a lot. So even going from right now, you open a catalog and you can find all these cool guys or fluorophores around. But at the time, like lots of them came from chemistry labs. So you have a team of chemists working on that. You try to put them onto your molecules. You try to image them, build the microscopes. You work with physicists. You work with chemists. You work with effectively like some people with a biochemistry background to frame the biology problem of interest. And you also work with folks, or we kind of try to scramble on our own and like kind of you try to learn everything together about data analysis. Right now, like you take an image and it's nicely image processed. At the time, it had to be done from scratch. So it was this attitude that it's an interesting idea. It's an interesting field. It doesn't matter that it hasn't been done before or that it's clunky. Let's just try. It's a really interesting question with a very unique approach that we are trying to answer. Therefore, let's just get it done. And I think that attitude of what is always really relevant, what is the most important question I can answer and how can I answer it? And then like really in both labs, both for my graduate student and the postdoc environment, it was always very important, I think, to all those folks that their people participate in all aspects of the project. So for example, even if you are joining a Steve's lab and if you're a physicist, which I wasn't by training, you had to go learn molecular biology and protein expression. Like people were not really expected to be handed out stuff and then like, oh, let's just go get it done. And it was same with Hardin McConnell. Like you had to really do things yourself. He really valued data, not just models. So that's something that you learn a lot. Like there is a model, there's a pretty cartoon in a paper, for example, this is how he would think this is work, but truly like, what is this based on? How did they make that the conclusion? How did you make that conclusion? And how solid is it? And combine that with this whole thing can-do attitude, like you need to go synthesize a peptide, go do that. Or you need to go purify or solve a protein, go and figure out how to do that. And that's the thing that teaches you this flexibility and resilience and I think kind of just maybe, as I said before, this can-do attitude, because once you're in a startup, you can't just like rely on the boutique experts and wait for someone. Maybe you can, that's what the CROs are for, but you kind of have to balance it out and you have to be comfortable knowing the different aspects of the process yourself. And I think that's one of the most things that came with working with all of them.
Those are some important characteristics, I think, for great advisors. They're mostly fearless. They're open to asking questions and trying to find answers to those things and encouraging their trainees to do the same thing. And fearless, just open the doors if they want to try something, and that collaboration with other people. I think that is the basis for being a leader at a startup. If you want to found a company, you need to bring all these things to the table. Am I missing something, or is that the gist of?
I think you really got it. That's a very good summary. Going back through like academia versus industry, to be honest, throughout my graduate school, it never occurred to me that I would not go into academia. Throughout my postdoc, it never occurred to me that I would, until the very end, that I would not go into academia. I was really focused on doing an academic work and doing what they call very basic research. And then I think kind of it was mostly towards the end, there were two factors that happened. One, it was 1 or 2 years. I watched my postdoc colleagues. It was a kind of bad hiring situation with both industry and academia, kind of struggle and go on market for academic jobs for 1, 2, 3 years in a row. I myself tried for 1 year. I went on 3 interviews, got 0 offers. And then I'm like, okay, I'll go try again next year. After that happened, the other thing that really more open towards was there was a postdoc run program that also started at Stanford. They were inviting people from industry, similar to what you are doing, but just in person. And then they would share their startup experiences, different from biotech. And I got more interested in that whole space. How do you actually start a company? And there was another colleague from W. Murner's lab who also went out and got his company off the ground. That style of thinking I was more interested in. How do you actually convert your skill sets from the academia into something that's very useful for people and maybe for the planet in general? And then it was just during that 1 year. And also at that time, but right around that time, I also met through a graduate student from W's lab. I met Pat Brown, who was at that time Howard Hughes professor of biochemistry at Stanford. And he was just starting a company that's now Impossible Foods. I talked to him. He offered me to join his initial team. And I just decided to go for it.
That's great. That was a nice segue into the transition from academia into your exposure to startup world. So you were recruited by Pat Brown to work for Impossible Foods. What were you doing there? Why did he hire you? And yeah, tell us about your experience there.
So I don't know exactly why he hired me. No, I should ask him that.
Well, let's talk to Pat.
Yeah, let me guess. I'm going to guess he hired me because I had this combination of protein chemistry experience and understanding of fat. I don't know if they're actually, when you kind of think of, okay, now Berkeley has a few people and lots of them are connected to Hardin McConnell in one way or another. There are not that many people who understand how lipids work or how the cellular fat works. And how do you interact with the proteins and thinking about both worlds together. And if you think about animal tissues, for those who are non-vegetarian, who are meat eaters, you have muscle, you have connective tissue. Mostly what we eat is a muscle tissue. But then that muscle tissue is interlaced with fat. For example, the most prestigious beef thing is called, I think some of it is Kobe beef, which is very interlaced with these little fat cells and fat deposits. And that would make things more smooth in your mouth and more flavorful because fat dissolves a lot of flavored molecules. So how do you kind of effectively engineer this tissue from animal muscle-like tissue, from plant proteins and fats? Is a question that Pat Brown was really interested in. And I think he brought me in another person who started with me. So she was a pure structural biologist. So she had this protein stuff really nailed down. She was actually, I think she had NMR and crystallography background. And then I think for me, I had this understanding of fat and proteins. But I think what Pat was looking for, and that's something I think everyone should kind of take a lesson from him, or maybe kind of go even talk to him or some of the podcasts. He assembled this team not based on experience in the field because there was kind of none. Sure, there were some people with food industry background as a part of the team. However, I think the core R&D team all came from very classic biochemistry, protein chemistry backgrounds in general. And with the folks who were interested in learning new things, very interested in the project, and could really think well about picking up different techniques, different aspects, different ways of thinking, and applying it to this new product that the company was trying to create. And I kind of was really, really inspired. And to this day, I'm very inspired by his style of hiring and how he thinks of how he thought about what type of people project really needed.
I'm going to reach out to you for introduction to Pat Brown. We need to bring him onto the podcast for sure. Talk about the work that you did and in the outcome. So how much time did you spend with Impossible Foods?
I didn't spend that much time, relatively speaking. I only spent around 1.5 years with them. But it was, for me, it was really transformative from the perspective. The other part from my being in these 4 really high-end, amazing advisors, those labs are really well funded. So you come out of the 4 labs or 2 labs, 2, 3 labs that are super well funded. You have newest, latest toys. Everything's available. You are at Stanford, like you're at Berkeley. Even if you don't have something, you probably go walk 2 labs down the hallway and you have a toy to use. And also, let's say in Axel Brunger's lab, I got used to working with these spaghetti-like proteins that are really hard, membrane proteins, kind of relatively speaking, hard to express, hard to handle with respect to how do you work with them, how you characterize them, how long they're stable, and things of that sort. And then you go into this burger place, and we use them to image single molecules. But you have this background of thinking through and how to optimize different processes. How do you keep these things in good enough shape? And then you walk into this food space where now it's really fast-paced. We have what we have funding-wise. We have some cool toys and other things we have to kind of make up on the fly. And for example, boutique biochemistry proteins, you can purify them based on the affinity tags that people put or different chromatography techniques. Now you have these plant proteins. None of them are tagged. They come from natural sources. And then the other part is hey, what do you do with them now? And then also for some of them, I can make this really elaborate purification process. But is that really scalable? So I think what really Pat introduced me to and working on Impossible is this cost analysis from early on. Yes, this is doable, but does it have properties that can survive scale up at the scale that we need? Number one. And number two, what is it going to cost me? And is that going to work? You cannot be selling $50 burger. You need to come to a price point. So at some point, you've got to start thinking about the process. If you pick a particular line of a process, is it too expensive? And I think kind of very early on, I think Pat was really good about outlining that and learning that and going from this small molecules, a very small number of things to look at and handle large number of them in a large quantity. I think at that place was the first time I made grams worth of stuff, because burger has to be a patty. You can see it with your eyes. You can stuff it in your mouth.
Yeah. Those are important lessons. I think that is what is missing in academia. Most of the time, we don't really have a sense of that cost because you usually have a grant or something funding your work. We don't know the real world costs of anything and that concept of scaling. We do everything in the micro scale. When you have to build a gram level burger of proteins and fats together, I think that is a real world application. I'm sure you enjoyed that experience. And I tell this to people, go work with a startup, early stage startup where you can put on multiple hats because you'll be forced to. There's not an option. Learning is not an option. And I think you'll learn tremendously from that. And from there, you went on to Eve Biomedical. And again, you're one of the first ones to join that company. You're pretty gutsy.
Well, Eve Biomedical was another. So I joined in Series A, and then they already had founders were in place. I got recruited to join Eve Biomedical through a network. So effectively, a friend of mine from graduate school, Stephanie Nishimura, she and I kind of effectively did this project with W. Murner and Harden McConnell together. If you look us up, we're co-first authors at the time when I think co-first authorship wasn't so popular. Not popular, just as a concept wasn't as well accepted. We really work really well together. Stephanie's an amazing scientist. And I really enjoy working with her. We work really, really well together. And when she said, hey, I know these folks from nowadays. And after her graduate school, she did a short postdoc and she went to Applied Biosystems. They got acquired by Life Tech. And then she knew a founder of Eve Biomedical. She used to work with him at Applied Biosystems. So she had this already kind of understanding of him. And she said, hey, they're trying to do something really, really neat. And at the time, it sounded really amazing. Effectively, what the goal of the company was to do a long-read sequencing by using a single particle tracking, by using polymerases as a motors, and then watching the single particle, this little bead, kind of run around. And by kind of doing some image processing, your one is to interpret the movement of this bead into a sequence of the DNA. And the main goal of their projects was to do long reads rather than short reads. And that sounded really appealing to me. It was this combination of a very, very cool scientific problem, like how do you sequence long pieces of a DNA? That was before Oxford at Nanopores actually made it. And then all these new workarounds and applications that are currently available. And also working with someone that I really, really enjoyed working with. And it was a really hard decision for me, to be honest, like leaving Impossible Foods. I had a fantastic time there, really great team, very vibrant atmosphere, and super important problem. Like the reason I joined Impossible Foods was I really believe in what they're doing. I really believe that one way to help our planet is by modifying our food source to help reduce greenhouse gas emissions. I'm so happy, and I've been so joyful to see them grow and succeed during the years. And it was a really hard decision, but kind of at that time, there was this really hard pull on almost emotionally was challenging because there is a point where there's this draw towards, I've spent last 10 years of my life doing these like single molecule assay methods. There's kind of a variation of it right now that people are trying to do DNA sequencing, which is a really important problem. And I have this one person that I absolutely enjoyed working with. And I decided to jump, but so you can blame it on Stephanie.
Let's blame Steph for this cause. But if you were to point one lesson that you learned working for Eve, what would that be?
It's really hard to be competitive. I think that they were in a really, really hard space. For example, if I contrast Impossible and Eve, Impossible is like effectively a front runner.
The first one, yeah.
The first one, the most innovative company in the space, complete trailblazer effectively in the space. To this day, not that I'm biased, they have definitely the best idea of how to move in that space. And I think the way Pat set up the company and the folks who were there to work with him and help push it forward, they are still the leaders, absolutely. Like just the way they think about problems and how he thought about problems. Now, Eve, in contrast, was almost more similar to the situation I got myself in Scalmi, which is how do you hatch something novel and survive in a very, very tough competitive space where there are lots of people around you trying to do very similar things. And I think that it's very different in a way, but that was something kind of merging both Scalmi's and Eve experiences together. You really have to move fast and you have to have results fast. Otherwise, your competition is going to catch up with you, not even in the ways that maybe you foresee going forward. And then it's really hard. How do you judge or how do you argue your own value to investors? How do you do that? How do you successfully raise the next round? What does it take to raise the next round? Like, how high is that bar? And the bar is not only you, the bar is you and all of the competition. So if someone else has done something in that amount of time that you had with the runway, if someone else has done something very different, folks could raise the bar for you higher than what you need to be. So I think that was the biggest lesson learned there, that once you are in a very competitive space, you've got to find a way to move fast, to move your product to a next value point. That's what you think. Now you have to kind of try to match it with what other folks who will be funding you or partnering with you will want.
Both are tough paths. Like, with Pat, he was the first in there. So you need believers, visionaries to fund that and help you move forward. And then when there's a lot of competition, moving fast is going to depend a lot on like funding and the talent that you have at your disposal. Both are important lessons. I want to end this academic discussion on, if you were to go back to grad school and postdoc and do something different, what would that be? Hindsight. Anything differently that would have helped you in the startup career that you pursued?
Yes, I would go and make friends with folks from different schools. So definitely on top of my list would be go to all these like law school parties and go to all the MBA parties and go to CS or engineering. What I've learned, like it's a joke, but it's not a joke. In order to raise a startup, especially, you're bootstrapping, it's a different story. Let's say you're given like your first check is like $5 or $10 million. Sure, you can go and hire the very competitive law firm. You can go hire yourself like accounting experts. You can go find yourself a business development person. But I'm talking about this like very, very initial idea when you're still like bootstrapping. At that point, you do need, and I think it's very valuable to have this loose network of advisors, not even official advisors, just folks that you can pick up a phone or send an email to and say, hey, I have this question. What do I do about this? And those are the people that really are valuable in a very small, just to give you a perspective. Let's say you're looking for a law firm first time around, like, who do I pick? I heard people use 1, 2, and 3 law firms in the Valley. Well, who would you go to? And how do you find someone? And I know, why would I want to work with this person or another? And then I think like having a person who can help you out with that, just from the perspective, even like if they offer you their advice, this is what I would look for if I were looking for a counsel, be a corporate counsel, or if it were a regular counsel, or you have to have your, let's say, some kind of financing, accounting going on. So how do you go find yourself an accountant if it's not some like ginormous big firm? Like, do I go to the biggest firm or do I go look for my boutique person around the corner? And just, I think having people from different disciplines, you can help them out, they can help you out, and just kind of leaning on that network is very, very important. So I think, and the other thing I would do, if you have any at all interest in entrepreneurship, I would go, I don't know whether like say, Haas School of Business has some of those programs. I know Stanford does nowadays, especially where they offer these like 2-3 months courses of, from the business perspective, what's important, like kind of like, what do you need to think about to build a business? And I think kind of just taking that like 2-3 months class or course while you're a grad student or a postdoc would be very valuable, just to kind of change your framework of how you think about problems. And also meeting some of those people, they can become great resource afterwards, both as a peers or someone who can become your co-founder or just come work with you afterwards. Jokingly, I think kind of more interdisciplinary, but not just in the science sense, but also kind of go move around the campus a lot and make buddies.
Those are great pieces of advice. I did that, not relate, because we tend to stick to a lab and just focus on the research. And spending 4, 5, 6, 7 years in grad school, we don't really form those relations. I think the best outcomes for your life are going to be when you form meaningful relations during your school days, and be curious and ask those questions, understand what other people are learning. And maybe you'll meet somebody like Steph that you would leave whatever you're doing and go join a company. I think people take networking as going to a meeting and like exchanging cards, but it's going to be about intentionality, about building a relation and learning about those people. If you don't need anything, you might be able to help them. I think those are great pieces of advice. Before we close with my traditional closing questions, what are your future plans and where are you heading next?
Now, well, I am right now with ALX Oncology. My next plan is to take 1 or 2 of our Scalmi programs and really see them, chaperone them through the clinic, at least phase 1 clinical trial. I really want to see that milestone. That is my personal milestone. I know like it's a long road towards anything else, but just getting there would be really fantastic for me personally.
Great.
I would really like to see that happen. And then we'll see what happens after that.
Good luck with that journey. And now to the traditional closing questions. If you have to share a couple of lessons from your Scalmibio that others could benefit from, it could be mistakes that you did or good things that happened that others could really benefit from. What would that be?
I would say going backwards, I would be more aggressive with seeking funding and not stretch out my bootstrapping for as long as we did. I think that's something that would really help out. Again, like that, I think that goes back to that whole concept of, if you look at both, let's say, Pratt Brown's or an Impossible Food style of fundraising, you've got to move fast. I think that's something that you hear a lot. And then I think I would say that I wish that we pursued more aggressively the partnerships or funding from earlier on to be able to kind of move much faster on that front.
Be aggressive with your bootstrap, with your funding, and don't bootstrap that long. I think that's a negative thing that comes from grad school. You live with little.
Yes. And you're used to like coming from grad school and a postdoc, you're very used to, I got used to, you get like, you dig yourself in your hole, you have your problem to work out. And effectively in a grad school and postdoc, right? Like you really got to work out your problem. It's always a question, the same question as before, like when do you stop? When do you quit? When do you move forward? giving that balance. A friend of mine told me, I think there's another great person I personally never talked to, but this is like a third-hand perspective. There's a professor at Stanford. I think he is in the chemical engineering department, and his philosophy to his grad students and postdocs is that you have to be very judicious with how you spend your time. And I think that's something that I actually haven't really encountered as much with my grad school and postdocs. It was more like work on a problem and get it done, as opposed to really be aware of the runway that you have. And I think that's something that is a very valuable experience for any industry, whether you go to a startup or a big one. You just have to be aware of the runway, like whatever that runway is, whether that's like physical, dollars in the bank, time to your next raise or whatever you have, and then like realize, okay, I need to make decisions in this time frame. I think that's something that lots of grad students and postdocs could maybe benefit from.
Wonderful, wonderful. Be judicious with your time and look out for that runway. And my next question is like, are there one or two people who you would like to call out that played a significant role for your success in the startup world?
Oh, absolutely. So it takes a village to raise a child, and it takes a village to raise a startup, even if it's such an early stage as this. And there was a really host of people. And I'll start with my co-founder, Jaume, who was such a pleasure having this journey with him. And I really appreciate all kind of his time and insights and desire and the kind of willingness to talk and hash these, like kind of flush these things through and help guide the project forward. Another person is, as I mentioned, is like Kirk Hansen. He is currently a professor at the University of Colorado. He is CEO of his own company, Omics Technologies, which is proteomics metabologist company. He was absolutely kind of also essential in actually securing SBIR funding. We were co-PIs in a grant. He kind of taught me about grantsmanship, or kind of this art of writing grants and how you think through this, and also kind of how do you think through problems. He's very, very kind of interesting scientist. Another person from a very different perspective is Mark Weinswijk, and he's a CFO. He currently and has been a CFO of several companies. And he's an MBA training, accountant by training. And he was a person who was really someone who you can turn to before you can hire a big, for example, accounting company or have an advisor. It's like just having him as an advisor to be able to call and say, hey, Mark, how does this work? If you can find a financial term or an accounting term or whatever, like it's just to have a sounding board to help you understand how do you run a company from a business perspective. And when you kind of have little questions, and he was just always so helpful, like, and such a great perspective, great practical perspective. And that is some kind of like, okay, you have this question or you have this problem, let's just go figure it out. And help me walk me through this really fantastic resource to have, and a person to just have. I don't know, it was really, really nice. Another person that was really essential for this whole process was Peter Straub. So he is a retired scientist. He used to have his own academic career. He ran a lab, and then he got recruited to a startup. He was in a startup that was like long time ago, maybe 30 years ago by now plus, and that got acquired by a series of bigger companies. And he retired as a high-level executive from Sanofi Aventis. In his retirement, he wanted a project. So he really stepped in and with this and was really eager to do effectively anything from bench work, planning, being a sounding board again, like kind of thinking through the problems, coming up with like experimental ways to do this. Very practical and very knowledgeable person who kind of understands also this whole process of drug development. And from his perspective, was actually more from a small molecules. His world was how do you develop a small molecule drugs? Another person, which was someone who was from very different walk of life, was Mike Beebe. And he is actually a mechanical engineer by training, serial entrepreneur himself. We're just good friends. And he's had such a super practical insight on how do you run a company. He has really, really interest in physically, how do you run small startups? How do you go get funding? How do you think through presenting your technology? And he didn't know anything about what we do, but just kind of not do like technically, but just kind of having him as effectively a person to talk to, like, I talked to this investor and this is, what they told me, or how do I emotionally deal with this? And it was really helpful to have his insights. And on the last folks I wanted to thank are my Morrison Foerster IP counsel. They really taught me a lot as to how to think about patent, what is patentable, what do you need to have to go through, like how do you think of when do you file, what do you file, what do you want, like how do you, what your claims need to be. So I really want to thank the Jia Zhang and Yan Kui from Morrison Foerster's Palo Alto office. They were fantastic to have in our corner.
Great. Marija, you've been very gracious in telling all the good things and failures to supportive about telling about both the sides of what happened in this journey. And I'm sure our audience is going to benefit. I definitely enjoyed this. I'm going to cherish a lot of these learnings and I look up to you in my future journey, but good luck with everything. Thank you so much.
Thank you lots of Naresh and good luck to everyone. We hope to see per Naresh's one podcast, at least 2 startups out there.
Absolutely. That's the goal here. Thank you so much.
Yeah. Thanks a lot.
Hello, everyone. My name is Hunter McDaniel. I'm the founder and CEO of Ubiquity. And our purpose is making an impact through materials innovation. We manufacture a new kind of quantum dot, which are tiny bits of material that are very effective at manipulating light. In the past, if you wanted to make a new color of material, you had to make a new composition of matter. But with nanomaterials, there's a new paradigm of their size and shape, and we're able to make essentially any color in the rainbow with the same composition of matter. They're used widely in the display industry today, but unfortunately, we're not able to go beyond that due to some materials issues that we've solved with a new composition of matter that we're making quantum dots out of. We've gravitated towards applications involving solutions to climate change, and we're deploying quantum dots into the facades of greenhouses as our first market, roughly a $4 billion per year market opportunity to enable growers to control the spectrum of light, the color of light in their greenhouse, and ultimately boost the efficiency of crop growth. When you put quantum dots into glass, you can enable a new functionality, which is electricity generation combined with transparency. The big vision there is powering cities of the future with the glass on the sides of buildings. And we've been installing windows in our first commercial buildings over the past year. That's about a $20 billion per year market opportunity. This technology was originally developed at Los Alamos National Laboratory and MIT, where we have exclusive licenses. We're also licensing technology from the University of Washington, and we have patents that we wholly own ourselves. Myself, I have a Ph.D. in material science and engineering from University of Illinois, and I worked at Los Alamos National Laboratory where the founding technology was developed before I founded the company some years ago. We're always looking for investors, so reach out if you want to support our journey, as well as customers and partners, and we are recruiting right now. You can email me directly at hunter@ubiquity.com or follow us on social media at Ubiquity or at Ubigrow, which is our greenhouse product brand. Thank you.
Thank you for listening to this episode of the Lab to Startup podcast. You can find links to the resources and programs mentioned in these episodes, connect with Naresh, or subscribe to this show at labtostartup.com.